clear regions are is they're areas where the bacteria were killed or destroyed by the virus. And so when we see a plaque, that tells us that the bacteria survived, or sorry, that the virus survived and it killed the bacteria that it was attacking. Now, with this in mind, then, that means that the nearby cells, the ones around those plaques or clearings, are also going to have been exposed to the virus. And so that means that we can harvest those cells and they should have virus inside of them. They haven't been killed yet because that virus is spreading out, but they've been exposed. So they should have a virus in there. So that's one of the ways that we can cultivate bacteriophages. For animal viruses, it's a little bit trickier. We have to grow them inside living animals, which again is an ethical concern. This is also really hard because, as I said before, viruses are very host specific. So if I want to grow a human virus, it doesn't always work to grow that human virus in something like an animal, like a cat or a dog, or even a chimpanzee. Sometimes they're just too different. And so because of that, in the past, virus testing a lot of times was done on humans, which again is a huge ethical concern. A lot of times then we can grow animal viruses depending on the strain in chicken eggs or in embryonic eggs. And the reason this works is because the eggs that are present, their cells inside don't have the same defense systems against viruses. So that's a good way for us to be able to grow viruses and cultivate them. For example, with things like the flu vaccine. If you get your flu vaccine each year, one of the questions on your flu vaccine card that you fill out ahead of time is, are you allergic to eggs? And you might have wondered, why do they ask me that? Like, that's nice that they're concerned, but why do they care? And the reason that they care is because the flu virus, when it's initially cultivated, is done so inside embryonic chicken eggs. They then isolate the virus from those chicken eggs, and then they are able to kill the virus, prepare it to be a vaccine. But there's always the chance that there could be a few egg proteins in there. I mean, even though they're very careful. And so that's why they ask about you being allergic to eggs for that virus, for that vaccine. The most common way nowadays that we grow human viruses is in what are called continuous cell lines or human cell cultures. What these are is they're cancer cells. So they're cells that were harvested or donated by humans that had cancer. And these cells then never stop growing. Remember, that's what cancer is. It's a cell that's stuck in mitosis. So these cells just keep growing and growing. They never stop. And we can keep cultivating them, moving from one petri dish to another. And because of that, we have human cells that we can work with that allow us to do research. Now, one of the most important people with continuous cell lines is a young African-American woman named Henrietta Lacks. And Miss Henrietta was actually the very first donor for a continuous cell line. She ended up having cancer, and you can read her story. I have it posted at the bottom. She ended up having cancer, and her cervical cancer cells were collected, and they are still used today. Her cells are still growing. And the reason for that is they're cancer cells. And so because of that, they're stuck in mitosis, they're immortal. So this over here on the left shows transformed cells, meaning cancer cells, and how they'll continue to grow and stack on each other. Normal human cells, like if I took a biopsy of us and tried to grow our biopsy in its cell culture, our cells that aren't cancerous, they only grow in a thin line and then they stop because we have contact inhibition. What that means is when cells start to stack, they realize that that's something that's dangerous. It could be cancerous. The DNA sends a warning signal, and so they stop growing. So they only stack like this, like what we see here when they're cancer cells. Now, Miss Henrietta Lack's story is a really fascinating one because her family didn't even know that she was a donor. And there's a lot of controversy around her cells, or at least there was in the early 90s, because they realized that Henrietta had never signed a consent form. Now, some people assume that this was because since she was African American, that the scientists just harvested her cells without her knowledge. And that really doesn't seem to be the case based on what we can find in the doctor records. Instead, it appears that the doctors working with Miss Henrietta were also studying cancer. And so when they took her cell sample, they decided to go ahead and keep the sample in the lab to continue studying and researching it. Later on, another doctor came in who worked with them, a colleague, who was talking about trying to grow viruses, and one of them suggested that they try using Henrietta cell line. They did, and Henrietta was the one who created our first cell line called the HeLac cells. Now, unfortunately, she didn't know, and she was never asked, and her family didn't know. When this came to light in the 90s, the medical groups immediately reached out to the family and asked if they could go ahead and have permission to use her cells continuously. And thankfully, her family said yes, which was very kind of them. But it's still a really interesting story. And like I said, I have it linked at the bottom because it's one of those things where, again, sometimes, doctors forget to ask permission when they're doing research because they just get so excited about their discoveries. So it's something that was kind of an eye-opener for the medical field too, to make sure that we're always, following the correct procedures nowadays. So how can we tell if a cell is infected with a virus? Well, one of the easiest ways to look for is to look for the cell changing. And we call that a cytopathic effect. And what that means is the cell is changed in some way. So this is what a cell line would normally look like. They're kind of spread out, they're stacked on each other, they're kind of smooth. And then this is what that same cell line would look like infected with viruses. They puff up, they swell, they kind of show that they have inflammation. And so if you take a petri dish of human cells and add a virus and look at it under a microscope, like a day or two later, this is what you would see if your virus grew. So that would show you that you have infectious virus particles. Another way you can tell is you could do a DNA test on here to see if the cells were infected. That would be your serological test. You could test for antibodies. We do that in patients. That's one thing we've been doing with COVID. We've been checking antibodies to see if people have been exposed to COVID. So that's another way we can identify the viruses people have been exposed to. And then we can do PCR. And that's what we do as well with COVID. We've been doing PCR tests when we're trying to figure out if a patient has COVID. So let's talk a little bit about how viruses replicate. And viruses, depending on their type, are going to have different replication patterns. Let's start with the bacteriophages first. So the bacteriophages are going to have two mechanisms. They're going to have the lytic cycle and the lysogenic cycle. And so what we're going to find is the lytic cycle is one that tends to be a short-lived cycle, meaning that they infect the cell and then they move on and infect a new cell. And the lysogenic cycle is where they infect the cell and then they stay with it. So the first step, the more common one, is the lytic cycle. And the way this works is a virus will attach to a host cell. So the first thing they have to do is they have to find a host cell that they're compatible with. They attach to it, and then they penetrate that host cell. Now remember, our bacteriophages kind of look like little spaceships. They have that little sheath. That sheath contracts, and it pushes DNA or RNA into their host cell. Once the virus gets inside of the cell, it starts to immediately make its enzymes. It starts going through transcription, translation, and we get biosynthesis. So if it's a DNA virus, we're going to have transcription. If it's an RNA virus, we'll just go right to translation. Oh, I love that. That is so cool. Next, they have maturation. And what this means is we've made our viral particles, but now we have to put them together. It's kind of like if you get a little box of Legos, now you have to put the Legos together to build whatever you want to build. And that's what the viruses do. And then they do a release. And what they do is they release a type of chemical like lysozyme that breaks open the cell walls of the bacteria that they've attacked. And that unfortunately destroys that bacterial cell. So when they get exposed to a virus, it is going to result in their death when the virus releases. The virus then goes out and it now looks for a new host. So here's that process. We have attachment, we have penetration, it's putting in its DNA or its RNA, whichever. We now have biosynthesis, which is where we make a whole bunch of copies of what our virus wants. So we make, the little particles, the DNA or the RNA, the little hexagonal heads, the little sheath bodies. Then we mature in there. So we put them all together. And then we're going to release out through the use of an enzyme like the lysozyme. Now, the lysogenic cycle is very similar. The only difference is that we have this process where the virus actually is going to integrate into the bacterial cell or into its host cell. So it starts off the same. We have attachment. So they attach to their cell. They penetrate. They put in either their DNA or their RNA. But then their DNA or RNA is going to become part of that new host. And so, for example, if it's a DNA, it acts like a plasmid. Remember, we talked about plasmids before? And so it's going to act like a plasmid and become part of the viral genome. Now, if it's an RNA, we don't really see this happen. So RNAs are rarely going to go through lysogenics. It's only going to be with our DNAs that can become part of the DNA. Then what's going to happen is they're going to stay there for a while. They can stay latent or in that bacteria's DNA for generations. So every time that bacteria cell replicates, it's going to replicate the virus DNA as well. It's just like a plasmid. And all of its future offspring have this little virus inside of them. And it's like a ticking time clock. They don't even know they have it, but eventually this little bomb is going to explode. What will happen is the virus eventually will decide to become active again. It will go through its process of transcription and translation, assembly, and then release and kill the bacterial cell. What we usually see is the virus stays integrated or hidden until it detects that the bacteria is getting old or weak. And we're still not sure how it does this, but it detects that something's happening where the bacteria is not happy anymore. And once it detects that its host is dying, the virus goes, well, it's time to abandon the cell. And so then it pushes forward, makes copies of itself, and releases and kills the bacterium on its way out. So it's kind of like abandoning ship. So here we go. The phage is infecting a cell. The DNA becomes part of the host DNA. It's like a plasmid. Now what's going to happen is every time this host cell divides, its offspring are going to get the DNA. When it becomes stressful, let's say the bacterial cell is about to die, the virus quickly will go through the rest of its process of doing its development, its assembly, its maturation, and its release. And then that way it can kill the cell and leave before the cell dies. It's a quick way for it to get out of there. Now, the reason this is important is when a bacterium becomes lysogenic, so in other words, lysogenic meaning that it has virus particles inside, it oftentimes makes the bacterium more virulent. And remember, that means more infectious or more dangerous. So what we find is sometimes bacteria can cause diseases that are a lot more dangerous than other times. And when we look into this, sorry, you hear my cell phone going off in the background. Sorry, I didn't mute for once. When we look into this, what we see is, for example, Corynebacterium diphtheria, the one we talked about that causes diphtheria last time, that it is much more infectious or dangerous when it has a virus inside of it. It grows faster. Streptococcus pyogenes, this is the one that we mentioned that causes strep throat. It can also cause another illness called scarlet fever, which results in like a raspberry tongue. You see at the bottom here, this bright pink tongue. It's like a raspberry tongue or a strawberry tongue because of the color. It causes pustules to form on the tonsils. It can be much more dangerous and much harder to treat. And what we found is that it usually occurs when the virus or when a virus is inside of the bacteria. Sorry, everybody. Can you hear me now? Sorry about that. Unfortunately, the electricity here is going in and out, so we'll just keep going. Sorry. So if I disappear on you, don't panic. I'll be right back. So sorry about that. Let me get my Zoom back up and we'll go with there. So you guys did great. You just wait for me and I promise I will come right back. All right. Okay, there we go. All right. Can everybody see the screen again? Are we good with that? Good. I'm getting thumbs up. Perfect. Okay. So I'm going to try to launch my poll again. And again, the poll is where you can click your answer and nobody knows who's voting what. So it's completely anonymous. Okay. So how do bacteriophages get nucleotides and amino acids if they don't have any metabolic enzymes? Do they rely on a bacterial host, an animal host, or none of the above? So go ahead and click your answer and submit it. Excellent. Good job, guys. Excellent. This is just kind of a neat way to participate without having to talk over each other, which is nice. Good. And when I get a few more people participating, I'll go ahead and share the answers. Again, they're anonymous. Don't worry. Nobody knows what you voted. So don't worry. You can, guess. You can just try. Okay, I'm going to go ahead and end it and share it. So here's our results. And if you voted A, you are absolutely correct. So if you voted A, you're absolutely correct. A bacteriophage gets nucleotides and amino acids from their bacterial host. Remember, by definition, a bacteriophage is not in an animal host. They're in a bacterial host. So the correct answer would be A. Okay, let's try it again. Vibrio cholera. Vibrio cholera produces toxins and is capable of causing cholera only when it is lysogenic. What does that mean for something to be lysogenic? So let's see if I can start my poll here again. Okay. Let's see if it'll let me go here. No. No, it looks like it does not want to let me do my poll. I'm getting frustrated. Come on, let me do my poll. Edit poll. Oh, well, it's not going to let me edit it. Well, it was nice while it lasted. Okay. You can we'll just talk about it. The bacterium has mutated. So do you think the bacterium's mutated? Nope, that wouldn't be good. The bacterium contains a virus. That would be the correct one. So the correct answer here is that the Vibrio cholera produces toxin and is capable of causing cholera only when it is lysogenic. And what that means is that the bacterium now contains a virus. So that was fascinating for us as researchers because we couldn't figure out why sometimes Vibrio was so deadly and other times it was harmless. We realized later it's because it really depends on whether or not it has a virus inside of it. And the virus just makes things worse. All right, let's talk about animal viruses now. So we've learned how bacteriophages can reproduce, but what about our animal viruses? Well, animal viruses, thankfully, have an infection process very similar to the bacteriophages. And it's nice because you don't have to learn anything special. The big difference is that they don't have to worry about a cell wall. So they're adapted to just kind of fuse with our plasma membrane. Their biggest concerns when you're an animal virus is being able to fool the human cell or the animal cell into taking the virus inside. So it's kind of like they have to wear a disguise or knock on that cell door or the cell plasma and try to get that cell to let it inside. And then they have to quickly infect the cell and replicate before the cell realizes that it's there. Because remember, we have organelles. And if our cell detects that there's a virus present, it will release its lysozyme and it will kill itself. It will actually sacrifice itself for the greater good. It'll protect its neighbors. So the virus has to work very quickly to try to fool our cells. So the process here is very similar to what we saw with our other viruses, the bacteriophages. They're going to have attachment. So they're going to have to bind to our plasma membrane. They're going to have to penetrate. Now, the way they do that is since they're not injecting through a cell wall, they tend to rely on what's called endocytosis. That's where our plasma membrane, if you picture our plasma membrane, it pulls in and pinches in and it pulls the virus inside with it. Once inside, the virus quickly uncoats as fast as it can, gets rid of its protein coat, and releases its DNA or RNA. Then we're going to have biosynthesis just like before. So we're either going to have transcription or translation, depending on whether it's a DNA or RNA. We're going to put together those little virus particles, and we're going to be released as soon as possible. So here's a picture of endocytosis. So here's our plasma membrane, and the virus has attached to it. It has tricked the cell. So now our cell thinks that it's something that it wants. It's, thinking maybe it's some glucose or some proteins that it needs. So it pulls the virus inside. And as soon as that virus gets inside, it quickly releases its deadly DNA or RNA, and it will infect the cell, and it will take over that cell. Here's another example showing it kind of in a cartoon picture so that you can really get an idea of what happens. Now, once the virus is ready to leave the cell, what it'll do is it will pinch or move through, again, that cell membrane. So sometimes the viruses will lyse the cells. That does happen with us. But a lot of times the sneakier ones, and this is so smart, they will instead push out of the cell. And when they do this, check this out, they will wrap themselves around with our own plasma membrane as an envelope. And that means now they can sneak around in the body undetected because they're kind of wearing this little mask that looks like they're one of our cells. It's very, very tricky. So I had a chat that was asked that said, I thought that this was, that our cells were smarter. How come viruses are able to get in so easily? Viruses are very tricky, and they're constantly mutating, so our cells don't recognize them as a danger. That's what happened with COVID. COVID was something that our cells had never seen before. And so when it showed up, it's kind of like somebody knocking at your door that looks perfectly harmless and you have no idea that they're anybody dangerous. Now, of course, we all know not to open our doors to strangers, but still, it's something that our cells are like, okay, this isn't tagged as harmful, I'll bring it inside. What happens then is as we're exposed to viruses or as we're exposed to vaccines, our cells are educated. And that's where I mentioned it's like a most wanted poster. Now, when the virus approaches the cell recognizes it and it sends out an alarm signal. It's almost like a burglar alarm where it alerts all of the cells in the area that the virus is present, and it tries to shut itself down so the virus can't get inside. So there's all sorts of little tricks that the virus do and tricks that we do as well. That's why we're not sick all the time. If it was that easy to trick our cells, think about it, we would always be sick. But it really is rare for us to get exposed to a virus and really get ill. It's kind of like it comes in cycles. And usually what that is our cells have forgotten and then they kind of remember, or the virus is mutated or something to trick us again. So here's a way that virus DNA or viral DNA can cause issues with our cells. And a lot of times what will happen is we'll have viruses that come in that take over our central dogma. Remember, our central dogma is DNA, RNA, protein. So when we get a DNA virus, the DNA of the virus comes in and it actually gets our own mRNA to read it. And then our mRNA will go to the ribosome and we'll start making viral proteins. An example of this is hepatitis B. Now, the reason that DNA viruses are easier to make vaccines for then is because of the fact that the DNA is more stable, but also because when we vaccinate ourselves, they recognize that DNA as being viral and our mRNA won't react with it. It's like our cell is much more cautious. And so because of that, it can't replicate. So that's one way that vaccines help is they knock out that strand of mRNA so we don't read the DNA virus. Now, as we mentioned, some viruses are RNA-based. So what happens here? Well, what happens is the RNA comes in, and if it is a positive sense, it goes right to our ribosome and gets read out. If it's a negative sense, it has to use our mRNA to make a copy of itself. So basically, our viral mRNA can't be read in the ribosome, so it has to use our mRNA to make a replica. And then our mRNA is now carrying virus information. It goes to the ribosome and it makes a protein. An example of a virus that does this would be something like Ebola. It's able to use our ribosomes to make itself, and it is going to be a negative sense strand. Now, the really oddballs out there are the retroviruses. These are extremely unique. And again, an example would be something like the HIV virus, the lentivirus that we mentioned earlier. These guys come in and they make a very unique enzyme called reverse transcriptase. And this is an enzyme that we do not make. It's an enzyme that only retroviruses make. And what this enzyme does is it takes the RNA of the virus. It uses this reverse transcriptase, which again is an enzyme that they create, and it creates what's called a cDNA. cDNA means a copy of DNA. Then this DNA is read by our mRNA and goes to the ribosome to make a protein. It's a lot of steps. It's very complicated. Now, the good thing about this is you might have heard that HIV numbers have dropped, or you might have wondered how it is that Magic Johnson or people like that who have HIV are able to still be alive after decades of exposure. And the reason for that is because with the creation of this enzyme, this reverse transcriptase, we have manufactured medication that blocks this enzyme. And so what it does is when HIV is, in a cell, we make it so that it can't make the enzyme that it needs to make cDNA and to replicate itself. So we knock this transcriptase out. This is great because we don't make it normally. We don't use it. This is something viral based. So by knocking this enzyme out, we don't hurt our own cells. What we do is we essentially put this viral RNA on hold. And as long as you're taking that medication, it's on hold for the rest of your life, as long as you're on those medications. So this is what is out there right now with different types of HIV treatments. So you can take medication that prevents you from having HIV replicate in the body. Now, we do know, though, that some viruses can cause cancer. So let's talk about how that happens. So what can happen sometimes, and this is still kind of a rare thing, is that sometimes viruses can get in our bodies, and unfortunately, they can start to impact our own DNA. Sometimes this is from inflammation in the cells. Sometimes this is from the virus just producing enzymes that affect our DNA. And again, it's rare, but what it'll do is it causes our DNA to become unstable. And so our DNA actually becomes an oncoDNA. And remember, we mentioned that oncogenes are ones that are cancerous. So it makes our cells unstable. And now they start going through mitosis indefinitely. Every time they replicate, then they create more and more cells, and this starts to build up a tumor. So we start to see the development of a cancer. And it's all because this virus was causing inflammation, or it was affecting our DNA and causing it to stress out. Now, I refer to a lot of these viruses as proviruses. And a provirus means that it's something that can, promote cancer. So it's something that we worry about. An example would be something like HPV. As you know, there's an HPV vaccine out there that is recommended for women up to the age of 25. And the reason for that is because HPV is a known provirus. If you get exposed, it causes continuous inflammation in the genital region, and it can result in sterilization when you get older, and it can result in cancer cells. So it's just something that, we recommend that people get vaccinated for. Now, it only works until you're up to about age of 25, because around that time, around that age, human fertility starts to naturally decline. And what we see is that if we give the vaccine to people that are older, the cells that it's actually affecting really aren't replicating that much anyway. So there doesn't really seem to be any impact. So it doesn't seem to help people that are older. It really seems to help people that are younger. And again, part of that has to do with our own natural fertility. So how do we treat these guys? Well, since the viruses aren't living, they can't be treated with an antibiotic. I can't knock out most of their enzymes. I mean, I could for my HIV because it had an unusual enzyme, but for most of the enzymes, I can't block them out because they're using our enzymes. That makes it really difficult. I do have antiviral drugs that I can use. And these antiviral drugs are going to be ones that can slow the replication of the virus down. And as long as I take these drugs within a short time frame of exposure, then usually they can stop the replication of the virus. Now, the problem with this is that it has to be a really short time, like within 24 hours of exposure. And most of us don't even know that we've been exposed that quickly. So this is why if you go to a doctor's office and ask them for, an antiviral, they're usually going to tell you no, because they don't, by the time you're showing signs and symptoms, it's already too late. Where we do see it more is if you have like somebody like a caregiver who gets ill, then they can give the AZT or the antiviral to their patients to make sure that they don't develop the illness. So hopefully that makes a little more sense in case you're ever wondering why they don't prescribe antivirals. Okay, let's talk a little bit about the different strategies the viruses have inside of our cells. So virus infections can have 3 different ways or 3 different growth curves depending on what kind of virus they are. And they usually tend to fall in one of these 3 categories. They can be acute, they can be persistent, or they can be latent. I just had a whisper. Would an antiviral work for COVID? So right now, unfortunately, COVID isn't releasing any enzymes that we can really target. And we're not able to really administer anything that is working right now for COVID. We've been trying, like with family exposure, and so far all of our current antivirals are not effective. That doesn't mean that they aren't trying. As a matter of fact, you guys probably saw that there's an anti-COVID pill that they're working on right now, and that's basically like an antiviral. And so they're trying, they're hoping they can get something like that on the market soon. They're entering testing right now at the FDA, but it's still under development. At this point in time, there's not anything that's available. But they're hopeful. They're hopeful with our research. We're still learning so much about COVID. So viral infections, then, they can either be acute, persistent, or latent. And each virus is going to follow this pattern, whether it's acute, persistent, or latent, depending on their own genetic material. So an acute virus has this typical pattern where they will infect a host. The host will show signs of illness, and then the immune system clears the host. So this is what we usually see. So we get exposed to a virus. The exposure has to be high enough to be able to infect our cells and get past our immune system. Now, this exposure, if it's high enough for the virus to get inside our cells, if we do a test right when we're exposed within an hour, we'll see a high number of viral particles. But if I was to give a person a test after 24 hours, I would see what we call the eclipse period, which is where I wouldn't really find viral particles in the blood sample. And the reason for that is because the virus is no longer circulating in the blood. It is now hiding inside of our cells. So this also makes it tricky to look for viruses. And this is one of the things why we have you repeat testing a lot of times for COVID, or you have to show that you have a negative test within a certain period of time. It's because of this eclipse period where sometimes you can test negative, but you really have the virus. It's just hiding inside of your cells. So that's why we have to be careful with our testing times. Okay, so the eclipse period, they're hiding. Then the virus replicates in the cells. It releases a whole bunch of copies of itself. And now we have what's called an acute infection. We show signs and symptoms. We're sneezing, coughing, sore throat, whatever the virus is going to cause. Then we show this decline as our immune system is able to get rid of the virus. And now we're clear and the virus is gone. So we see this, for example, with influenza. We see this in most cases of COVID, where we get exposed, we get sick, and then we clear it. Now, we can have some viruses, though, that are referred to as persistent. Persistent viruses are ones that last for a long period of time. And so what happens here is we get exposed to a virus, and we think we've cleared it, but unfortunately, we haven't. And the virus is basically inside of us still, and it never really was completely killed off. It's just kind of living there, kind of hidden. And it may or may not come back later. So the host cells are still infected. They have this virus inside of them. Every time our host cell makes a replica or a copy of itself, its daughter cells have this virus, and the virus is just kind of sleeping inside. Now, with a persistent infection, the key here is that it can suddenly become active at any given time in our life. And this usually doesn't coincide with stress or with any kind of immune system drop. So it's just a random issue where suddenly the virus becomes active, and we're still trying to figure out what it is that's triggering that virus. Usually we'll see it become active when people are like stressed normally, but with persistent viral infections, that's not the case. So suddenly the virus becomes active again. When it becomes active, it starts to replicate in the cell, and it can come back and cause an illness that might look entirely different from the original virus. A great example of this is measles. So one of the things I hear a lot of times from people who don't want to get a measles vaccine is that measles never killed anybody. Technically, that's true, but measles doesn't always clear. And what that means is if you are exposed to measles, that you can carry measles for the rest of your life. Now, you may never see it again, but for some people, the measles may reemerge. When it does, it usually comes back as something called subacute sclerosing panencephalitis. If you hear panencephalitis, that should strike fear in your heart. Encephalitis is swelling of the brain. Pan means all of it or everywhere. So panencephalitis means that your brain swells. So subacute sclerosing panencephalitis is where the measles virus has moved to the brain. And again, we don't know what it is that causes this to happen in some people and not in others. It's most likely some sort of a genetics variation. For people who do get SSPE, it is a death sentence. So if you all of a sudden have your measles virus reemerge as SSPE, usually the patient does not survive because of the massive brain swelling. So technically it's correct. You're not dying of measles, you're dying of SSPE, but it's from the measles virus. And again, it's not something that happens all the time. So it's a very weird reaction, and we're still trying to learn about it. So I have a question, is this the same thing that skin diseases like eczema? So eczema a lot of times is not always necessarily a virus. Sometimes that's genetics, where our skin is producing too much of a dermal tissue, or it's producing too much of an enzyme or a protein, and that causes our skin to get too thick. So it looks kind of scaly or itchy, and it becomes irritated. But the persistent viral infections, those are usually ones that will be like I said, they're kind of like measles. They're pretty rare. They're ones that just never really go away. They're just always kind of there, but we don't know if it's going to come back. Eczema, I would say, since it's something that kind of comes and goes, is actually more like a latent viral infection, which is the one we're going to talk about now. If we wanted to compare it to a virus, I would put it more in this category. And this is one that's actually more common with viruses. And what a latent virus is it's one that comes in, it causes an acute infection, and then what happens is it permanently changes the host DNA. So that can be through inflammation, or it can always be in the cell, and it can come back anytime you have a decrease in your immune system. So let's talk about an example. An example would be something like herpes. If you ever get a cold sore once, you are now prone to getting cold sores for the rest of your life. Why? Our bodies, for some reason, cannot clear the herpes simplex virus. And so now it's always slumbering or always present in our cells. Anytime our immune system drops or we get stressed, that virus pops back up because it detects that our immune system is dropping, and it starts to cause mischief and it starts to grow again. That's why a lot of times we see people get cold sores when they're stressed. Same with genital outbreaks of herpes. Herpes can come and go. You might have an outbreak, and then all of a sudden you won't have any outbreaks for a while. It just depends on stress levels and whether the virus can come out. Another one would be something like chickenpox. So chickenpox can come back later. What does it come back as? Does anybody know? What does chickenpox come back as when we're older?
Shingles?
Excellent. Shingles. Well done. Yeah. So chickenpox is a nasty little virus that if you get exposed to it naturally, now if you get the vaccine, you don't get this, but if you get exposed to it naturally and recover, the chickenpox virus goes into hiding at the base of our spine. We actually can see it in spinal tissue. And it hides in there. And then when you get stressed out, it follows the nerve endings from the base of the spine through the body. And a lot of times it'll attack along the torso region, the torso nerves, and it causes shingles. Now, the reason we tend to see it in people that are older is because their bodies have forgotten what the chickenpox virus looked like. And the chickenpox virus just kind of waits and then reemerges when it thinks our cells have forgotten it. This is why you get booster vaccines. This is why you get shingle shots. There we go. When you're in your 50s, it's a booster to remind your immune system what the virus looks like. But if you were exposed to chickenpox as a child naturally, you never get rid of it. It's always hiding inside of you. These are also going to be ones that a lot of times can be associated with cancers. Now, chickenpox is not associated with cancer, so don't panic. But herpes can be, HHPV can be. So we do, worry or try to watch patients that have had these kind of infections. So the big difference, the key difference between our latent infections and our persistent infections is both of them stay in our bodies. The persistent infection is rarer. And when it comes back, it doesn't come back for any rhyme or reason. We don't see because of a loss of immunity. We don't see it associated with stress. It just comes back suddenly. And we think the difference is because of our genetics. Something happens that triggers it to start growing again. We're still not sure why. With latent viral infections, these are more common. And these are ones where they go into a dormancy period, almost like an endospore. They stay asleep inside of us. And then they come back anytime that our immune system drops or we have stress in our life, then they come back. So I just had a question. If you get chickenpox when you're a baby, are you not able to get that vaccine to prevent it? So they still recommend, if you got chickenpox as a baby, a lot of times they'll still recommend that you get the vaccine just as a precaution, just to make sure that your body's immune system remembers it. But if you choose not to, you have the same immunity because you were exposed naturally. You produce the same antibodies. So you wouldn't have to if you got it naturally. But you would still want to get a shingles shot when you become older as a reminder. So that way, because our immune systems start to forget. 5 decades is a long time to remember things. I can't remember what I did yesterday. So 5 decades, come on. So that's why we have boosters.
Professor?
Yes.
Sorry, would that be the same as so with the COVID vaccine, if you did get COVID and you do have the antibodies, but they still recommend you to get vaccinated, does that make much sense?
It does. And the reason for that is because the strain of COVID that you got might be different than what's generating right now or what is present right now because of mutations. So we already know that we have COVID Delta right now, and that is very different than the original COVID strain. So if you got COVID originally, there's no guarantee that the antibodies you have right now will work against Delta. And that's why they're recommending the vaccinations.
Okay, thank you.
You're welcome. Yeah, COVID is it's very easy to mutate. It's very similar to what we see with the flu vaccine. And that's why we recommend flu vaccines every year, is because they're constantly mutating. And so even though you had flu one year, you'll get exposed to a different strain the next, and then you could still end up getting sick. I have another question in the chat that says, do you have a higher chance, where'd it go? Do you have a higher chance of getting chickenpox again as an adult if it goes hiding in your spinal tissue? So you wouldn't really get chickenpox again. It would come back as shingles. So once you get chickenpox, you have those antibodies for the rest of your life. And you get those antibodies either through a vaccine or through natural exposure. Chickenpox doesn't really mutate. That's why this works so well. But we still have to give a booster vaccine to make sure that your body doesn't forget what the virus looks like. I have another question. So cancer depends on blood sugar for energy. So that means staying away from sugar. Ooh, good question. So what they find is that cancers, and these are ones that are not necessarily associated with viruses, these are just cancers in the body, that cancer tends to grow faster when there's high blood sugars. It's almost like cancer cells. So if we picture cancer cells undergoing mitosis constantly, mitosis requires a lot of energy. And so if I feed the cell a lot of glucose, it will be able to replicate even faster. So that's the correlation between sugar and cancer. This is why they tell you if you're a cancer patient to try to eat less sugar, or if you have cancer in your family to try to eat less sugar. Because if you do have a cell undergoing rapid mitosis, if you're giving it sugar, you're feeding it so it can grow faster. All right. So hopefully that answers those questions. Good questions. Okay, let's go on and talk about viroids and prions. So viroids are going to be things that are found in our environment that are pieces of RNA. And these tend to affect plants more than humans. And this is something that can affect, for example, the growth of vegetables or fruits. And what they are is they're where a plant maybe got exposed to an RNA virus, the plant dies, and it could be from old age or whatever. And the RNA virus that's inside of the plant will get released into the soil through the plant deteriorating, whether it's dropping its leaves that might have the RNA virus in it or in its roots or whatever. Now these RNA viruses are in the soil, and when they're in the soil, they are considered to be viroids. And what that means is they're just pieces of the RNA virus that have kind of fallen apart. Now, if I have a plant growing nearby, it can pull these viroids in through its roots, and it can end up getting infected or sick. This is one thing that can also affect our potato plants. We talked about the potato famine. Viroids can affect potatoes, for example. Here's normal potatoes on the left, and then these are viroid potatoes. Now, if we were to consume these potatoes, we wouldn't get sick, but they wouldn't taste good, not because they have a virus inside of them, but because they don't have the normal amount of starch. So the potatoes would be a little bit bitter. They wouldn't have that normal amount of starch and sugar in there. And they also wouldn't be the right size. So, it wouldn't be something that would get us sick. It's just something that is present in the environment. Prions, let's talk about prions. Prions are infectious proteins that were first found in the 80s. And one of the diseases that was associated with prions was mad cow disease. Prions are baffling to us. They're basically a protein that has been folded incorrectly. We talked about, baseball gloves and oven mitts before. So instead of getting a baseball glove, it's like I folded and then I got an oven mitt. But the difference with a prion is this oven mitt now goes and bumps into a baseball glove, and it converts that protein now into a oven mitt. And then it bumps into another baseball glove and converts that one into an oven mitt. So in other words, when you get exposed to a prion, it is a mouse-shaped protein that then bumps into other proteins and causes them to change their shape, and they become incorrect. We're still not sure how this works, but it happens. We also cannot kill prions. If you autoclave a prion, it doesn't kill it because it's already denatured. It's already deformed. And so if we try to denature it again, it just refolds and it bumps into other proteins and it still causes them to refold. These are the oddest things we've ever seen out there. They can, unfortunately, get into humans, and they can get into us through ingestion. So if we ate a cow that was infected with mad cow, then we potentially could ingest prions and get ill. Through transplants. So if somebody has, these prions in their body and they're in their organs, and we transplant that organ into a new patient, then they too will now have prions. Or through dirty surgical instruments. So that's why so many of our surgical instruments are now disposable. We get rid of them. Some of the diseases out there I mentioned are mad cow disease. Another one is Creutzfeldt-Jakob disease. That was named after the researchers who found it. And then the last one, Kuru, is a brain disease that's the same as the other two. But what is different about Kuru is it's associated with cannibalism. So they found that there was a tribe in Papua New Guinea that would celebrate the death of a loved one through ritualistic cannibalism. So if somebody passed away, the partner of that person would consume their loved one's heart to symbolize their love. And their oldest child would consume their brain to symbolize the passing of intelligence. And another person might consume their eyes to symbolize that they will now be the visionary in the family. So it's very ritualistic. And so what happens is that if they had somebody who had prions in them, they were exposed to prions through this cannibalism. And so in this tribe in Papua New Guinea, they knew that Kuru existed, some sort of like, odd disease. But it wasn't until researchers started taking samples of people after they passed away that they realized that it was related to a prion disease. That does seem to be a link here. Usually with prions, they're associated with, people eating people, like cannibalism. Mad cow disease was actually from cattle eating cattle. And I know that sounds weird. The cows were not, trying to eat one another. It was a human thing. We were taking cow parts that weren't selling. They were being ground up and put into grain as a filler. And, cows aren't supposed to eat other cows. They're not built to do that. And so we think that led to the mutation of the prions is, when you have a vegetarian, basically, when you have a strict animal that's not supposed to be eating meat and you're giving them meat, it causes this weird mutation. So what do prions look like? Well, here's what they look like. Here's normal brain tissue, nice and pink and normal. And here's brain tissue with Kuru, mad cow, and Creutzfeldt-Jakob disease. And so what happens is you start to get what are called plaques, these big clearings in the brain, and they affect memory, they affect motor skills, and they will eventually lead to death. So I had a whisper, somebody direct messages me and said, so you don't survive disease with prions? And the answer is no. It's just a matter of time. There's different times as to how long it takes for the brain to develop this much plaque tissue. But once it's present, then there's not any survival. And we don't know how to stop its progression. So prions are very scary. Thankfully, they're extremely rare. We don't really see prions very often. When we do see them in animals, we immediately cull or remove those animals, and we're constantly testing for them. So we have not had a case of mad cow disease here in the U.S. We're very careful with how we import our meat. We're very careful with animals being brought in to try to prevent prions. Okay, let's talk about our virus families. So these are going to be examples of some of the main virus families that are out there and ones that you've probably heard of. Yeah, what's up, Brianna?
I was wondering, so you were saying if they find it like in an animal that they pull the animal, but what exactly do they do since they can't kill them? Like, how do they?
Right. So usually what they do is they cull it, and then they will burn the animal. So they have to destroy it. But you're right. I mean, the prion, the burning, usually in a fire usually seems to get rid of it. But I mean, like an autoclave doesn't work because it's already been, mutated and it's going back into another cell. When you're burning something, there's not really anything left to be consumed. The only way it would really be exposed is if somebody ate the ashes, which, that isn't going to happen. So what they usually do is they dig holes, they burn the animal, and then they bury the animal. And the prions don't seem to be spread in water. It seems to be like it has to be inside of a tissue to be spread. Does that make sense?
I think. Thank you.
Okay, so like if you're consuming the burned ashes, maybe you could get exposed, but since they're buried, it's not like you'd really find the ashes to consume. And then if if that soil gets washed away with water, if you drink it, it's not really, the prions really don't seem to be transferred in water. It has to be like we're eating the tissue of a mad cow, or we're eating the brain tissue, or not the brain tissue, but the tissue of a human, or we have to involve some sort of digestion or an organ transplant or blood exposure. Like the prion has to be actively inside of other tissue, if that makes sense. Is that okay? Good. Okay, good. Okay. So let's talk about some of the main groups then that are present. And thanks, Brianna, for raising your hand. By the way, if you chat to me, I always try to keep an eye on the chat room, but if I miss you, you can always raise your hand and ask me, or just ask me at the end too. But yeah, raise your hand and I'll get to you right away. I don't want you to think I'm ignoring you, because I'm not. I'm trying to get all your questions. Okay, parvoviridae, this is referred to as fifth disease, and you might have heard of this. It's also sometimes called slapped-cheeked baby syndrome. And what it causes is it causes these red cheeks to appear on children. And we usually see it in toddlers or children under the age of 7 or 8. And we do sometimes also see it in immunocompromised patients. We don't usually see it in patients that are older and healthier because our immune systems are able to get rid of it pretty easy once we get older. It's called fifth disease because it was the 5th most common illness in children when it was first named. So that's why they call it fifth disease. Now, parvoviridae is not the same parvo you hear about with dogs. That's the first question I want to make sure we answer. So this is not parvo in dogs. Parvoviridae is a non-envelope DNA virus that will cause bright red cheeks, high fever, and rash in children. And again, it's something that we usually see associated with kids. You might have heard of it if you have kids. Papoviridae. Papoviridae is also a DNA virus, but it's unenveloped. And it is the one that's responsible for HPV. And this is genital warts. So there's more than 100 different varieties out there. And some of them, unfortunately, are linked with cancer. Because there's so many strains, we can't have vaccinations for all of them, which is why people still do get HPV, even if they have a vaccine in place. The vaccine gets rid of 15 strains, the ones linked with cancer, but there's a lot of varieties out there. Again, it's one of the most common STIs that's transferred, and we average about 14 million new cases every year. Here's a picture showing the oral HPV, and again, it's just like little wart tissue. The way to treat it is you have to go in and burn these little warts off, and that will get rid of the infection, for a little while. But a lot of times they can stay hidden in cells and it can come back at different times. Poxviridae. These are going to be envelope DNA viruses, and these were the ones that were associated. These are the ones that were associated with smallpox. So we saw these guys earlier. They also were associated with cowpox. That's how we were able to get rid of our smallpox. They also have a virus associated with them, which we still see today, called molluscum contagium. And this is a picture of a child showing molluscum contagium. It basically looks like little barnacles. That's why they called it mollusk. It looks like little barnacles on the skin or little bumps on the skin, kind of like a little bug bite, but it just never goes away. And so at first, a lot of times they're thought to be warts, but they'll do a sample on them and realize it's a pox virus, and they can be treated with antivirals. But again, it's just kind of a slower process because they have to go in and lance or get rid of the lesion on the surface, and then they can treat the inside with an antiviral so it doesn't come back. Herpes viridae. So these are going to be our herpes strains that we know can be latent. So again, these will go into hiding and they'll come back anytime our immune system drops or decreases. We can have HHV1. We talked about how we can have ones that are more with the oral, that's HHV1, or more genital, HHV2. But again, there's multiple strains of herpes. And again, that makes it really hard to be able to come up with any kind of vaccine because it's constantly mutating and changing on us. Envelope DNA viruses. Another example would be hepatitis B here. Hepatitis B is dangerous because it can attack our liver and it can cause liver failure. Now, thankfully, because it is a stable virus, we were able to produce vaccines for it. You usually get exposed through contaminated water or contaminated food. It is rare here in the United States because of the fact that we have so many safety issues in place for food and water. So it's not something we see spread here. But if you were to travel overseas, it would be more of a risk. So this is one of those vaccines that CDC recommends if you're going to be traveling out of the country, because your chances of encountering it in another country are much more likely than what you would see here. Picanaviridae, this is one that's associated with poliovirus. And so this is a virus that attacks our nerve endings. Now, it's an RNA virus, but luckily for polio, that virus tended to have a lower mutation rate than many of our RNA viruses. So we were able to come up with a vaccine for it. And so far, that vaccine seems to be working. It's holding up well, which is great. But we're always checking it because we're always looking for new variants. There's also another illness out there that's recently emerged that's called acute flaccid myelitis. And this affects the myelin sheaths of the nerve cells, and it causes the cells to not be able to function well with the passing of neurology impulses. And so what'll happen is you'll have patients that will start to complain of having like tingling in their arms, and then they'll wake up the next morning and their arm is completely paralyzed. It's very similar to what we saw with polio. We don't know much about it. It is considered to be an emerging infectious disease, and we're still trying to learn about it. It unfortunately does seem to affect children mostly. We have not had a case here in California, so we haven't heard of it here. Right now, it tends to be more in the Midwest. And for some reason, it also tends to be seasonal. We see it in the summer and not at other times of the year. And we're trying to figure out what it is where, it's causing this virus to kind of have this cycle. And that's what you see in that graph there, is kind of that cycle showing that it's really active in the summer months and then gone during the other months. And we're trying to figure out what it is and how it's spreading. We don't even know how it's spread at this point. So we're still learning about it. Rhinovirus, that's our cold virus. And again, that's one that mutates very easily. And we finally just gave up on trying to come up with a vaccine for the cold virus. We're like, it's not deadly, deal with it. So we just kind of gave up. And then hepatitis A virus. So this is another one that is recommended to get a vaccination for if you travel overseas or if you travel to a different country. And it's very similar to hepatitis B, where it impacts our liver and it can be spread through water and food. Coronavirus. So we've been hearing a lot about coronavirus. So as we know, this causes an upper respiratory infection usually. We see that with COVID. However, in some people, it can result in stomach upset and others a sore throat. So it's really hard to know what exactly is causing the issue. We originally saw our very first coronavirus in the early 2000s affecting humans, and it was called SARS. And that was sudden acute respiratory syndrome. And again, it started in China and it stayed in China. It didn't spread to the US, so it never became a pandemic. We lucked out. The reason it didn't become a pandemic, though, is because it was actually deadlier than COVID, and it showed signs and symptoms much sooner. The problem we ran into with COVID is it took a full 2 weeks a lot of times for people to show signs and symptoms, which means they were spreading the virus that 2-week period and they had no idea they had it. With SARS, if you were exposed, you showed signs or symptoms within 48 hours, if not sooner. So it was easier to isolate patients, and we didn't see as many carriers as we do with COVID. With COVID, we have a lot of people who don't show any signs or symptoms ever, and yet they're still sharing the virus. With SARS, it wasn't like that. If you had it, you knew you had it. And you would usually, be hospitalized, and it did have a higher death rate at that time than COVID. Enveloped RNAs. So these are things like our Ebola virus, our Marburg virus. I know I'm running out of time here, so we'll probably stop here in just a minute. This is a picture of Ebola, and this is a patient who has recovered from Ebola. But the reason I show this here is this white part right here is the retinal nerve in the eye. And these green regions are scar tissue that was left behind from the Ebola virus. So the Ebola virus, for some reason, loves to travel to the eye, and it loves to follow the optic nerve. What this means then is this patient who is showing this scar tissue is developing what's referred to as an Ebola cataract. And so Ebola, even if you recover from it, can negatively impact your vision. So you'll now have lifelong vision issues because of all the scar tissue that Ebola leaves behind. So it's quite, damaging. We'll stop here. I won't cover this slide. We'll just go ahead and stop here. So we'll pick up here next time. We'll finish out this chapter. And then just as a reminder, we do have our quiz on Wednesday, just like our normal plan. So please plan to be in person. The storm should blow over by tomorrow. I think it's supposed to actually be gone by tonight. So we can be back in class together. So please plan on attending in person for your quiz. For lab today, yeah, I like that. People are happy about that. Yeah, this is an odd thing to Zoom. But like I said, I didn't want you guys on the road. I didn't want to be on the road. I wanted all of us to be safe. It's just too dangerous today. With the with the quiz, so make sure you're studying for the prokaryotes and eukaryotes. It'll just be on prokaryotes and eukaryotes, no viruses. Okay, we're not having any viruses on our quiz. You have your homework due as well, so don't forget about your homework. Get that turned in. And then for your lab today, you get to watch a video and then answer questions about it. And I know videos about microbiology sound boring, but I've had people tell me this one's an interesting one. It's a well done documentary. And then you'll answer questions on it and submit those questions. You have until Wednesday at 1 p.m. to submit that video question information. All right. So I'm giving you a couple extra days just in case your power goes out. Yeah, it's a good video. All right. So with that, you guys have a great day. Stay safe and stay dry.